Howdy, my name is Seth Murray, and I'm going to be presenting on behalf of the JR Biotech Foundation for African scientists. I've previously recorded 2 videos on molecular plant breeding data analysis and methods, part 1 and part 2. That's on my YouTube channel. I'm going to plan on adding this one to the YouTube channel as well. And here I'm going to talk about going from molecular genomics to phenomics and phenotyping for quantitative crop improvement. So I'm not sure everybody's background here, so just to get everybody on the same page, we have had in the US, which is where I work in Texas, a tremendous increase in yield thanks to technology. Now the estimates are that about 50% of that's due to plant breeding and about 50% of that is due to agronomy. And we can see the various crops within the United States. On the x-axis here is the years of production. And on the y-axis here is the US scaled 5-year moving average of yield. And so this is the amount of crop that's produced per unit of land area. And we can see maize has increased about 9-fold in the United States. Potatoes have increased even higher than that, but most of that's due to agronomy. And then we have sorghum and rice and barley, wheat, beans with various lower levels of improvement. So yield is the thing that most breeders have traditionally concerned themselves with. And the way we got here was by domesticating these crops like maize. And here's a picture of the wild seeds of maize, which look like little rocks in the wild plant, to something that looks like this, which is a commercial hybrid. We started beginning that process 7,000 to 9,000 years ago with domestication. Up until the 1900s, primarily in the United States, regional heirlooms were grown. So these are open pollinated populations with very little scientific genetic improvement that are just allowed to open pollinate. And my understanding is that some regions in Africa are still growing open pollinated type populations because of seed challenges or seed system challenges, or the amount of money that farmers have to purchase hybrid seeds. In the 1930s to the 2000s, we really got into a phenotypic and scientific breeding era where individual companies and individuals were scientifically starting to select for improved yield in both hybrids and inbreds. And in the late 1990s, we started getting into marker-assisted selection and biotechnology. That was aided quite a bit when genomics came through. And now we've shifted largely in industry and elite material to genomic selection today. Now I mentioned yield, but I don't want people just to restrict themselves to thinking about yield only. Because I think if we've learned anything over the last 50 years from the Green Revolution to what's going on in the United States, that we can't just keep our mind on yield. We need to think about reducing input use. Right? We have a dead zone in the United States from certain areas where either fertilizer is over-applied or where it just leaches through the soil before it can be utilized by the plant. Human health and nutrition. So we want not just high calories, but we want something that's going to be nutritious for the people consuming it. If farmers aren't profitable, then their operations are unsustainable. So we have to make it so that any improvements we make benefit the farmer to keep them in the system. Ecosystem service provision. We want healthy water, we want healthy air, we want healthy soil. And so we want wildlife that can create a resilient ecosystem to prevent us from large pest outbreaks, for instance. We have to have some type of climate change resilience going forward. And increasingly, consumers in the United States, and I think everywhere, are looking for improved flavor. So this isn't really important for the overall concept of the talk here. I just want people to be aware that we can improve all of these traits through breeding, just as we improve yield. Now I'm going to use the case of maize. I am a maize breeder primarily. And maize is a great case study. So this is part of the international collection held at CIMMYT in Mexico of the tremendous diversity that exists within maize. So we somehow want to find the genes or the alleles in these different varieties and these different cultivars and these different races of maize to use them to improve our crop for whatever trait we care about. And it's important to keep in mind that genetic diversity is the fuel for this. So if we only have one inbred line, for instance, it's almost impossible to make any type of genetic improvement except through some type of mutation breeding. So we really want to screen breeding programs in the germplasm bank at the right level to extract information that we're interested in. So this is just an example of what maybe all the genetic diversity of wild and cultivated wheat looks like. Now, once we went through that bottleneck of domestication, it's going to reduce some of the genetic diversity, right? And then within any individual breeding program, they're using a subset of that genetic diversity. So in breeding program 1, for instance, they may have some of the same material as breeding program 2, which may exist in different regions throughout the country or throughout the world. And they may have different material than in another breeding program, which I should have labeled 3 here. Then even within a breeding program, we might have this larger source of material, and then we have a source of elite material, and then we might have a specific biparental population we created to do for future selection. So it's really important to understand this, whether we're using molecular tools or phenotypic tools, what level we're coming in at, because we're going to reach different conclusions. We're going to have different confounding factors. Phenology, flowering time, plant height, all these things are going to affect our end conclusion. So we need to just be aware of what level of genetic diversity we're talking about. Now, if we make an elite by elite cross, the pace is really slow by most people's standards. So I'm a Samsung fanboy. So my Samsung S9 Note was 7 years removed from this BlackBerry. Now that is a dramatic increase just in terms of every available feature in this product. When we compare that to a new variety of corn, that's all we can get in 7 years. So it takes 7 years to develop and release a new variety of corn, right? And why? We have controlled conditions we can develop these technologies in. They're made in very high production, high technology production facilities, whereas we have the environment to deal with. We have lots of confounding variation we need to somehow identify and select for in maize. So we can reduce this down to the breeder's equation. And so hopefully most of the audience has seen the breeder's equation before, but again, I just want to get everybody on the same page. So the breeder's equation basically says the genetic gain or the progress we're going to make over time has to do with the selection intensity. So the more plants we can grow and the fewer we can select. Selection accuracy. So can we measure those plants better or find the best plants more accurately? The additive genetic variance. So can we use more of the genetic diversity and select more of that? And then the years per breeding cycle. So anything we can do to speed up the years per breeding cycle, we're going to benefit from. And so one of the ways that we've sped that up is through molecular tools. So breaking it down to individual markers that we can use in selection before we even put those plants in the field. This slide's a little bit out of order, but we also need to have these larger programs to screen more material. And so I'm just using the example again of what I know, which is Texas A&M University. And these numbers differ a lot from year to year. And I think this was 2016 numbers. I just did an informal survey of my colleagues. And we're growing 10,000 plots for breeding, and then another 17,000 plots for yield trials. And we're measuring those with a combine. We're measuring those by hand. It's very labor intensive work. And then we look at something like cotton, which has much larger plots and they have a lot more to control for. And they've got thousands and thousands of plots, right? And so when you look across this, what you see is if you have multiple breeding programs together, there might be an economy of scale you can leverage there. So we can do that with genomic tools to some extent by buying a sequencer. But one of the things I hope to convince you of is we can do that with phenomics tools as well. Now I showed you the yield increase in maize across the United States. I just want to point out that in Texas, in my environment, it's a little bit different. Now, the reason this is important is because I suspect for most of you, your environment is a little bit different than, say, the largest breeding program in your state, in your country, in your continent, right? And so what's happened over the years is that in the US, industry has consolidated and moved to the Midwest, and they're selecting increasingly mega varieties that cover larger areas because of the economy of scale of seed production. But what's happening is more marginal environments like Texas, which has a lot of heat and drought stress and very different photoperiod, is being left behind. So another way we can address this issue is by doing more trialing, right? We want to get at genetics by environment, by management interactions, and then each of those individual main effects. And so I'm going to talk a little bit about data from the Genomes to Fields project, but this would be something I'd encourage all of you to consider as well, is having these large multi-cooperative trials. And I know CIMMYT and many of the internationals have excelled at this for a long time, where everybody is growing the same or similar material, so it can be analyzed jointly, and you can really try and get at the underlying basis of variation that's useful. So I mentioned molecular genetics, and that was the focus of my previous talks. And it's just important to remind everybody that, it helps us grow more plants because we can pre-screen some of this material through DNA. It helps us measure plants better because molecular markers are very heritable. However, the traits that those molecular markers help control often don't have that same level of prediction. And it also helps us create more seasons per year, right? And so what this example here is at the bottom is just a Manhattan plot, they call them, from a genome-wide association study. The different chromosomes are on the x-axis, and then the significance of the markers. And this is just for some large Mendelian traits. This is actually a published study that my former student Adam Mahan and Stephen Anderson and some others and Trish Klein helped put together. And so we identified the gene for red cop and yellow and blue grain, but it was more of an example of how you might do this. Okay, let's take a step back. And I think I presented this slide in the molecular plant breeding talk. So there's been three major waves of genomics. And these three waves, I think, are going to be very predictive for how we use phenomics tools in selection going forward. So let's just walk through this real quick, which is in the mid-1980s, QTL linkage mapping using biparental populations and genetic markers like RFLPs and AFLPs became a popular scientific method to try and understand the genetic basis of quantitative traits. And the idea was that we'd be able to use those in plant breeding. And what this allowed us to do was make specific inference to specific populations. A lot of knowledge and publications were generated from this technique, but I don't think we can really point to too many examples of where there's been substantial crop improvement. In 2005, based on some of the output of the human genomics community, we came up with association mapping as a scientific community of plant breeders here. And the goal there is to find loci and find genes, just like in QTL linkage mapping. But instead of creating these with biparental families, we're using diverse populations. We're finding alleles that work across population structures. And this has been rather exciting to find things that are more broadly applicable. And it's, again, helped us generate a lot of knowledge and publication, but you don't see a lot of people actually having made impact with crop improvement. So if the goal is crop improvement, GWAS and QTL linkage mapping haven't really delivered on the promise that was always claimed. In 2009, with Mewis and et al., we started to see genomic selection and some of Rex Bernardo's work and a lot of other people's work. Genomic selection came on the scene. And the whole philosophy behind genomic selection is different. Instead of trying to break down scientifically and dissect the genome and find something we can select within the genome, the goal is actually to predict and select the best individual. And so we're doing that by assigning all the markers some type of a function instead of just finding a marker that's significant, which gives us a lot more power to find the best individual, right? And we can use that across different type of populations. It's very predictive across training germplasm, and then you apply it in a cross-validation scheme and see how predictive it ends up being in a real-world population. And what we found from this is we can actually get a lot of crop improvement. Most of my industry colleagues are extensively using genomic selection, genomic prediction. We've generated a lot more knowledge about genetic actions in the genome in crops, but it's really hard to publish on this because there's not a model that other people can take and deploy. Right? And so we've gone from this idea of discovery or estimation of loci to prediction to try and find things that we're interested in. It's a lot easier to include value indices. So if you want to select for both yield and sustainability and health and taste, you can do that, although diminishing returns with every other trait you want to add to your selection matrix. So I present that genomics because genomics is 30 years ahead of where phenomics is. Oftentimes, phenomics is presented as an afterthought to genomics, but really genomics is a sort of mature field at this point. It's delivered what it always promised to deliver, which is helping us make better decisions in the breeding process through genomic selection. Genomics is a bottleneck. I'm sorry, phenomics is a bottleneck for genomics, but I think phenomics also has a very promising future in itself as well. And that's what I'm going to talk about for the rest of this. So high throughput field phenotyping is the act of going through and measuring as much as you can, or as many plants as you can, using usually some type of imaging or remote sensing technology. Now, it's very similar to the idea is very similar to phenomics. The idea of a phenome is measuring all or many of the traits across plants, and that can include over time. So a lot of times people think about these technologies like drones, which we can see at the bottom there, unoccupied aerial vehicles, UAVs, or unoccupied aerial systems. We try not to say unmanned because that tends to be seen as a gendered term, but drones or UAVs or UASs. So those can work. These ground vehicles. So this is a ground vehicle. I'll show you a picture at the end that we built for clearing mature maize, and it's loaded full of sensors. And a lot of what people have so far talked about doing, and I'm going to talk about it first here today, is to automate routine measurements. So measuring plant height in the field is really labor intensive. So if we can use these technologies to do it when it's 40 Celsius out there and nobody wants to be in the field, we might be able to get more data and make better decisions. We're also finding that it can estimate grain yield in those crops where the grain is actually visible. So in sorghum or wheat or things with a panicle that's sitting out. Corn has a tassel. The ear is actually below the canopy, so we can't really image that so far. Cotton's another good one. Cotton has actually had very good success with estimating cotton yield from UAV imagery. We can also potentially use this to estimate disease, and there's been a few papers on this now. So you can start to see disease either through, I'll say this term multiple times, RGB or red, green, blue. So it's what's in your cell phone or in your standard camera. You can see a lot of diseases through that. Then we can also talk about multispectral or hyperspectral imaging. And that usually involves a totally different type of sensor getting into the near infrared spectrum. So these are all things that we can automate that are currently manually collected. And that is a huge win. The next level, though, is to find new signatures of eliteness. So I can't have a crew go out and manually measure plant height every day on my crops, but I could have a UAV out there every day measuring plant height on my crops, right? So then we can start to connect these temporal growth patterns and get biomass estimates. We also can get spectral signatures. So it's very hard with a backpack to get a hyperspectral signature of a plot or a plant, whereas it's very easy with a UAV. And I'm going to show you a couple examples of all these things in green. So from there, the next leap, both of these things, though, are similar to QTL mapping, right? An automated routine measurement. GWAS, where all of a sudden we can get a little broader. We can find some new signatures of eliteness by looking a little bigger, right? And then the last one is phenomic selection. And this is directly comparable to genomic selection in the way that we do it. Except instead of using genome-wide DNA markers, we're using phenome-wide phenotypic markers. And I'll get to this at the end. One of the real exciting things that can potentially come out of all of this is if we do identify new signatures of eliteness or stress signatures, perhaps those can be used for farmers, because farmers can also access these UAV tools. And to me, that's something that's really exciting for the applied or practical outcome. Overall, these are going to help us grow more plants better, measure plants better, use more genetic diversity, and create more seasons per year. Oh, this slide looks a little wonky. I apologize. But I told you I'd talk about plant height. I'm going to just start off with the case study of plant height because my group has spent substantial amount of time looking at plant height. So one of the interesting things in Texas, now this doesn't apply to most of the United States, but in Texas, plant height, even in elite commercial material, is highly correlated to yield. So what does that mean? It means that taller plants tend to yield more. Now there is a bias that taller plants shade out shorter plants, but we've done enough analysis now that I'm confident that that's not the majority of what's going on. I think what's going on is plants that can grow through our stress of heat and drought and the other pests that we have, abiotic and biotic stresses, tend to be more vigorous and yield higher. And so you can see in this study here, which was published in Field Crops Research in 2013, we had 14,000 individual observations on traits like plant height and yield in commercial material, and the correlation across Texas was extremely high. Then similarly, I mean, this is just one of many genetic mapping populations. This is a biparental mapping population we had in test crosses, and plant height and grain yield were also highly correlated. Okay. So right now, we only measure plant height, or right now as being like 5 years ago, but we only measured plant height by using this ruler. And so we can, measure this plant height, say this plant is 102 inches if we measure the tip of the tassel. So we're getting what we call terminal plant height. What we hypothesize, though, is if plant height is really predictive of an ability to avoid stress, there may be a lot of things that we're missing in between. So if we have drought stress periods, maybe one variety is better at drought stress at early growth, and one variety is better at drought stress in later growth. And we could never see that with the conventional methods. The conventional methods that we use to collect this data. Sorry, just wanted to make sure I was still recording. Okay, so our outstanding colleagues, Soren Popescu and Malamba Lonesome, along with a lot of other people who I'll thank later, did this great job of comparing their structure from motion, which I'll show you a little example of, collected from UAVs, so that's on the y-axis, to our manually measured height. And so what we could see is that in different plantings, that correlation is a little bit different, but still we have very high correlations between what we measure with a ruler and what they measure with the UAV. Now we can look at this slice across time and see this was measured in May 13th and June 3rd and June 16th and then July 1st. And we can see that the correlation is pretty good all throughout, but then right here it falls apart a little bit. And what that was there was a difference between the day we measured with the UAV and the day we measured with the ruler. And the plants were growing so much that those correlations sort of fell apart. Okay, so what's better? So a lot of people would think that the ruler is better, right? And so how do we actually get at that? What do we know the truth of the true plant height being? So one of the ways that we've really looked at this is by using the genetic variance components. And any plant breeder, I don't know why so many of these slides converted funny, I'm sorry. The plant breeder is very used to measuring genetic variance, okay? And all this is we have multiple replicates in the field. We can dissect the variance into things like genetics. Oh, I don't have the other legends on here either. I think one of these is replicate variance, one of these is environment variance, and one of them is G by E variance. And we can see that the genetic variance increases over time. So one of the ways that we look at this is at different height measures. So we have 2 manual measures, ligule and flagleaf, and then we have 4 UAV measures we extracted from the images from structure from motion. And we have the max, which is the very top point. We have P90, which is the 90th percentile in the point cloud, P95 and P99. And so we can see at some points, actually, this is the genetic variance, okay, the percent variance explained by genetics, and the data measurement. And we can see at this point, the UAV measurements actually did better than our manual measurements. And then when we get to the end of the season, the manual measurements, legal height is the most repeatable measurement, right? But look at how many more measurements we have from the UAVs. So this is a different tool for measuring plant height. And once we did this, and this is a very small study, once we did this, we actually found what we would call crossover effects. So what we measure at the terminal plant height, traditionally with a ruler, we never would have seen everything that happened before that. And there were times throughout growth that other plants were the tallest plants. So for instance, this purple plant is the tallest till here. And then by the time we go to measure at the end of the season, it's no longer the tallest plant. And so if height is correlated with yield in your environment, then understanding this could be important. So that's plant height, but we want to scale this up. We don't want to just measure plant height as an academic exercise. We want to develop a system that we can really get better answers to our breeding questions. And so if you are interested, this was published in PLOS One in 2016, Shi et al. Very nice paper where we talked with the whole group. I think at the end, there were about 40 faculty members who were all working together to try and make this happen. And so we had aerospace engineers, electrical engineers, computer scientists, we had the field researchers such as myself, ecosystem scientists like Malambo Lonesome and Soren Popescu, and ag engineers, all coordinating all of this. And of course, it required some funding, and so we had some administrators. But I'd say of everybody, the ag engineers are really the key to make this all work and to bridge these different groups. And so this is some examples of some of the many people who are involved here. But I would not have understood this process if we hadn't worked in this team. And I think too often as plant breeders, we tend to just stick with what we know and just apply what we know, instead of actually reaching out to other disciplines to see how we can get more together. So I'm going to go through these steps real briefly here, and then I'm going to show you some examples of how we go from the UAV data to the field. I hope to just show you that plant height data so that you get a little excited about learning the details of this. So the first step is we place ground control points in the field, and I'm going to show you what those look like in a minute. Then we fly a UAV or UAS to collect the pictures, and you need a GPS and an inertia measurement unit tag on those. And so this, we've tended to move to DJI Phantom 4s for most of our RGB imaging. And that's simply because they're very inexpensive. They collect very high quality data, and it's very easy to process and fly the drone. So I can actually have college students who are flying the drone and collecting data for us on these things. Then we need to download the images. We need to do some type of spectral correction if we're going to try and calculate a vegetation indice. We have to orthorectify and mosaic the images. And I might as well explain that now. When in your phone, if you ever take a panorama, there is an algorithm that stitches the image together. And that's really important because if you just took a bunch of different pictures of a field, you wouldn't know what plot you're in. So by creating these orthorectified mosaics, we actually can tell what is in each picture. And that's really important because otherwise from the drone, you wouldn't know how to connect your plot to various data sources. We then make 3D point clouds and do structure from motion. From there, we start identifying the plots so that we can actually say what's in them, take the measurements from them. We need to identify the ground in the structure from motion and separate that from the plants, extract the data by plot, and then we're going to import that into some type of statistical software and run statistical analysis. So all of this procedure, I actually, really didn't know how to do and still probably couldn't do by myself. It's only through the talented team members here and my awesome graduate students that this has been possible. But the exciting news for you as scientists is I think these tools are sort of on the cusp of becoming able to be routine. And so once you've done all this, which seems like a lot, but if it's like genomics and software comes out to simplify your life, it probably won't be all that difficult to collect. Then we can get the data we need to make better decisions. So I mentioned ground control points. So this is an example of our ground control point. It's a piece of metal welded on a metal pipe and set in concrete in a bucket in the field so that we can remove it for when the tractor comes. And we've placed these throughout the field. Now this really helps with stitching this orthomosaic. So I don't know if you can see the fuzzy edges here, but there's probably 500, 600, maybe even a couple thousand pictures that make up this orthomosaic, okay? And Stephen Anderson was one of my former grad students who did a great job assembling a lot of the information I'm going to present to you today. So what does a point cloud look like? So when we orthomosaic these images, we can actually, just like a stereoscope, by taking the image of the same plot at different directions, we can get its 3D structure. And so structure from motion generates these three-dimensional point clouds. And so we call these point clouds because these are literally X, Y, Z points. Stephen then was able to classify the ground points, right? Those are the lowest elevation in the field. And then you can extract those using this LAS tools and figure out what's the plants. Okay, and so this is a 3D representation, a structure from motion of plots of the plants in our field. And then Stephen looked over growing to degree days or over days after sowing the crop, how the manual height on the x-axis and y-axis correlated, and it was very strong. Now here's the problem, or a problem anyway, is that oftentimes as you're mosaicing this if your data is collected poorly, if there's too much wind, if you did not have enough overlap between images, if you were flying too fast, if your camera sensor wasn't, properly set up, you can get these black holes in the image stitching. So that just means the algorithm could not stitch together those photos. It's like the panorama, if somebody's moving it, it stretches them. We have no idea what this artifact is. This was always a very interesting one that came from the 3D point cloud. And then here we have the vast majority of the plots, but then the algorithm calculated that the ground is up here and the ground is down here for some of these points. So obviously a lot of problems. And those are still sort of typical. And so we've done some work and other people have done some work. So I'd encourage you to read up on this, is to figure out how to optimize your mosaic. What's the optimal flight path, for instance? Do you want to fly back and forth? Do you want to fly back and forth and then crosswise? How do you do this? And so there's actually calculations you can make to figure out, with the resolution of your camera, what height do you want to fly and how fast do you want to fly. You can also have problems with images over or under saturating. So you really want the color to be between the maximum and minimum value that the sensors can pick up. And then we can also have these problems with blurring. So it's hard to see individual plants in this case. So I mentioned ground control points for spatial recognition. One of the things we've learned is that we need to have some color differences. And so that helps with the spectral calibration. And you actually need to go through and use GPS to figure out where each of those are. And then the last problem we had, or the last major problem that we had that I'm going to talk about, is how to actually identify the plots. So when you go into these UAV tools, most of them have been set up for engineers or not plant breeders who deal with thousands of plots. And so at first we had to manually draw boxes around these plots to say what we wanted to extract data from. So one of the things my student Stephen Anderson did was take some rough code I wrote and actually make an R package. And so this actually takes, and I'll show you in the next slide, takes a breeder's field book. And as long as you have the two coordinates on the side of the field and the size of the field, it will actually draw these boxes for you and connect to your Excel spreadsheet with what's in each of the plots. And so it'll make what's called a shape file. So your remote sensing colleagues can tell you what a shape file is, but each one of these boxes that's over one of these plots has what the variety is and where it is in the field, and then that can be useful for extracting the data. And so what Stephen wrote is called RUAS tools. And so if you are collecting this type of data, once you mosaic it, this is a great package in R to help you actually extract or develop the shape files so you can extract what's in them. Okay, so that's sort of the nuts and bolts of how you fly and how you collect data. I'm going to go back to some of what we discovered and get you excited on the path to phenomic selection. So I mentioned the Genomes to Fields project. So we've grown the Genomes to Fields project for many years, and we tend to try and fly two types of vehicles, a copter vehicle and a fixed wing vehicle, about 2 times per week over the 500 plots in the field. And so we did various irrigation treatments, and there's about 250 genotypes times 2 replicates. Okay, so one of the things Stephen was able to find by comparing these different platforms, and then by comparing a late planting, a optimal planting, and a late planting, is he was able to find different patterns between these various plantings and different patterns between the platform that was used. And so you'll see this noise in the Phantom 3 Pro. A lot of that has to do with where the vehicle thought it was in space. And so you can see all the plots all of a sudden got taller, and then they got shorter, and then they got taller again, which is an artifact of this type of data analysis. Now, we don't see this, or we haven't seen this in our Phantom 4 Pro, so it could have been an error for that year. This is still fairly experimental. We can also see that different varieties have these crossover effects, and The tallest variety, for instance, in the early planting and the late planting is this blue line here. And the blue line here in the dryland planting is buried somewhere in there. Right? So we see different growth effects of these various hybrids. Now, Stephen also used this in a genetic mapping population. So remember, we talked about QTL linkage mapping populations, GWAS, and now genomic selection. This is a biparental population. And in this case, or 3 biparental populations. And then in this case, you can actually see the 2 parents, and then you can see the transgressive segregation of their progeny. Now, some of this has to do with these are not completely homozygous inbred lines. They're still, I think these were like F5s. So there's a little hybrid vigor there. But in this case, you can see that there's not as much transgressive segregation, and you can see the differences between the various recombinant inbred lines. And so this is day after planting on the x-axis, and then the plant height measured by the UAV. Now, one of the coolest things when we combine this with genomics information is we actually saw that the QTL change over time. So remember, normally we measure plant height with a ruler. And if we only measure plant height with a ruler, we would only detect the QTL that show up at the end of the season. But by measuring plant height with a UAV and using an algorithm to sort of smooth that data to calculate plant growth for all recombinant inbred lines, Stephen's actually able to show that some of these QTL fade in by day 50 and then become basically irrelevant by the end. And so there's different genetic control over plant height at different times segregating within a population that we can take advantage of. Okay, so I'm going to switch gears a little bit and talk about Natalia's data set. And she used the Genomes to Fields project as well. But what she did is put us on the road to phenomic selection here. So she had a total of 532 variables. She wasn't just measuring plant height. She was measuring different vegetation indices. And then within those vegetation indices, she's calculated different canopy cover versus mean plant versus mean plot. Her background was remote sensing, so she had a good handle on how to do all this. And so she extracted 28 phenotypic features times 19 flights, and that gives her 532 variables. And then what she did was she tried to correlate these variables or use these variables to predict yield, right? So this is very similar to genomic selection. In genomic selection, we're going to use hundreds or thousands of markers spread throughout the genome. Here, she's using hundreds of phenotypic markers spread throughout the phenome to predict yield. And this was a very, very exciting result because what it showed me was with the data set she collected, we were able to predict the top 10% of yielders. Now, when we actually look at that, though, that's a lot of data to process. 19 flights and flying all that time and processing the data. So I challenged Natalia to reduce that to a fewer number of flights and see how good she can do at predicting yield with that. And this is what was really exciting to me. So again, predicting the top 10% of yielders, the R-squared isn't great, but we don't really care about predicting the lowest yielders, right? We throw those away in a breeding program. We don't care about the middle of the pack. We care about predicting which ones are the best hybrids out there. And so what she was able to find was with 3 flights collected in May and June. This was well before flowering, NGRDI canopy cover, a little bit after flowering, NGRDI plot standard deviation, and about a month after that, NGRDI plot mean. And she was able to predict the top 10% of yielders. Now, why is this so exciting to me? Well, we haven't even run the combine through the field by this point. We still would wait another month or month and a half before we got the yield data on this. So perhaps it is going to enable us to make breeding decisions earlier, which is going to speed up that cycle of breeding decisions. Now, when we tried to look at what's actually going on here, so on the x-axis is days after sowing, and on the y-axis is NGRDI. This is just a vegetation index. It's like similar to NDVI, if you're familiar with that. What she saw was really interesting when we reduced it down to just a few varieties. So here's 3 commercial varieties. These are presumably unrelated. They're from 3 different companies in the US. And then here's 4 hybrids that all share a common parent, TX714. And what we can see is that the NGRDI signature looks much more similar for these 4 than it does for these commercial checks. And so probably what's actually going on is that the NGRDI, the vegetation index, doesn't actually predict yield itself. It's just helping capture the relatedness between these individuals and predicting which individual is going to do best. Okay, but you can see this is kind of noisy. And what that has to do with is when it gets windy or when tassels come out, tassels are not green, they tend to be yellow. And so they're going to change what the vegetation index looks like. So I just told you about plant height, and then I told you about how we could use temporal data, and then I told you how we could use lots of temporal data to make better breeding decisions. I want to go back to something fairly simple that that I think everybody could conduct or grasp. This was an opportunistic study, thanks to Gary Oddbody, who planted my elite hybrids in his field. We have a disease called southern rust, Puccinia polysora, and we also have one called charcoal stalk rot. Dr. Oddbody, a plant pathologist, was growing our yield trial in his location. And I happened to come down and notice that there was a little bit of disease. And so I, just asked him, well, when did this happen? Can we screen it? And he's got enough of his own work. He doesn't need to be doing my work. So he went out there and started screening, it from then on. But we missed all that early, plant disease. Well, it just so happened another colleague, Michael Sterick and Jina Jung, both, were collecting UAV data over these plots. And so what we were actually able to do was go back and look retrospectively to see when the disease started and how it emerged. Okay, and so to me, this is like the Wayback Machine. So if anybody's familiar with this, it's an archive of the internet. And you can learn more about it at that website. They don't do anything with plants. It's just they archive old websites so they're not lost forever. So now I have a record of how my plants looked in 2017, even though I never stepped foot in that field. And we can see here that there's a little bit of rust starting. Okay, so this is the first day I think I got there. And Dr. Oddbody rates this from 0 to 10. And he said this plot was an 8. And this plot, which happens to be one of mine, was a 1.5. And this plot was an 8. These 2 8s are commercial checks. And this is one of my experimental hybrids. And then you can see the disease progression here. My plot's still really happy and green. And these guys are getting almost completely covered in that rust. And then charcoal stalk rot moves in. And Dr. Oddbody scored that as well. And because my plants weren't stressed from the southern rust, the stalk rot didn't move in. And so these plants are completely dead at this point. Again, I never stepped foot in the field, but I can see now how that disease progressed. Okay. And then when we go, we go back 2 weeks later because it was too windy. So that's one of the downsides of UAVs is you can't fly in the wind. We can see that these are starting to dry down now and these are completely dead. Okay. So that's just an example of another opportunistic thing. Even if you don't have any data science skills and you don't have any remote sensing colleagues that can help you, even today you can use these archived images for learning something about your crop and making better breeding decisions. But there is a cost, and that cost is not really the UAV because the UAVs that we're using, a Phantom 4, can be had in the United States for somewhere in the neighborhood of $1,000 to $3,000, depending on the options. One of the bigger costs, though, is storing the data. So with our fixed wing aircraft, once we have the raw images plus the processed images and the point clouds, that's 14 gigabytes for flight for like 2 hectares, about 1,500 2-row yield trial plots. When we have the rotary copter, we got about another 11 gigs of flights. I suspect that's quite a bit larger now. Our ground vehicles, though, because they're not collecting images, they're collecting proximal sensing, was much smaller. Okay, so this data really can add up if you don't know what to do with it. So one of the things we did was we actually came up with a data structure, and there's an organization called Cyverse, funded by the National Science Foundation, that helped us figure out how to make this data publicly available and fair, or findable, accessible, interoperable, and machine readable. And so you can look up this. I mean, it's not a formal publication. It's got a DOI. You can look this up, but this is a data set from 2017, and we got one from 2018 and 2019. And you're welcome to freely download it, play around with it. If you can make a better decision than we did with the data, because there's yield data in there too, that's all the better. I mentioned the ground vehicle. So the UAV is what I'd highly recommend for all of you. UAVs are fairly inexpensive. They're easy. We spent a lot of money building this thing so it could clear mature crops. And then we basically found that it takes too long to go through the field. So by the time we start at one end of the field and go to the other end of the field, the lights changed. Everything about the plants has changed. If it rains, it gets stuck in the mud. It's hot, it's noisy, it's dirty. Nobody likes being in it. It takes 5 hours to do what the UAV does in about 30 minutes. So I recommend generally against ground vehicles and to focus on the UAVs. But we still were collecting a lot of different types of data from this ground vehicle. Now, I'm not going to talk about this. This is not my work, but there's an organization called ARPA-E, or the Advanced Research Project Agency for the Department of Energy in the United States, that funded Agriculture's Hubble, they call it. And so this is a giant gantry system that runs on these rails right here. And this is all steel. And this tower comes up and down. And inside of here is a whole suite of sensors. So not just RGB cameras, but thermal cameras, hyperspectral cameras, lots of different things to sense the crop. And the reason I wanted to show you this is to show you the possibilities of where we're going to go. So RGB data is really easy to collect. It's fairly straightforward, but there's all types of new sensors that are coming online that we could add to use and make better breeding decisions going forward. Okay, I'm going to change gears just a little bit to try and get us to phenomic selection. So I want to talk first about near-infrared spectroscopy. And I'm going to assume that a few of the people who are watching this have done near-infrared spectroscopy, but most of you probably haven't. So I'm just going to run through how this works. And this is based on solid physics. And it's the idea that in the near-infrared spectrum of light, certain chemical bonds absorb and give off different signatures. And so what we do is we scan our corn sample, we then grind it, and we scan it again using this near-infrared spectrophotometer, near-infrared in reflectance mode. So this cup of corn kernels rotates around, there's light shown on it, there's a detector, and this is a Fourier transformed NIR, a thermo-antares. And that absorbance and reflectance spectra, the reflectance spectra in this case, is measured. And so the various spectra of our corn samples look like this. And on the x-axis are the spectra, the actual spectral data point, and on the y-axis is the reflectance value. And so you can see the different corn samples have different patterns. And we don't try and interpret anything from these patterns. It's completely impossible and doesn't make sense. What we do is develop partial least squares calibrations between the reflectance value and the wet chemistry of this corn. And we can use that to predict starch, oil, and protein. So we'll build a calibration from, let's say, a couple hundred samples. And once we've built that calibration, we can apply it to thousands of samples. That's like identifying a genetic marker for high oil. And then we can go into a germplasm bank or a breeder's collection and screen a lot of them for oil. Okay, we're doing the same thing except using light and near-infrared spectroscopy. So one of the things, again, I want to show you is the power of genetic variance components. So what I'm showing you on the x-axis here for both crude protein and phosphorus is the actual wet chemistry measurement. On the y-axis is what was predicted from NIR. And we can see for our calibration samples in blue and our validation samples, which are the purple crosses, crude protein looks so much better. And when I showed this to all my engineering friends, they say, well, crude protein is a much better calibration. However, again, in breeding studies, we have lots of replicates and we grow them over lots of environments. And so when we actually put this into our genetic structure, our experimental design structure, we actually found that there's less error in the phosphorus predictions than there is in the crude protein predictions. And the genetic variance is about the same. So from my perspective as a plant breeder, this phosphorus calibration is every bit as good as the crude protein. And if we look at the range, just because this could be a trick if you didn't know this, the range for phosphorus is similar to the range for crude protein. Okay, it's about a twofold range. So how can we use that near-infrared spectroscopy data? So we happen to have this data set where we used NIR to predict a GWAS panel under dry land and irrigated samples. And in 2018, this wonderful paper came out by Rinson et al. Called Phenomic Selection. And they used NIR to predict complex traits in wheat and poplar and showed it was as accurate as genomic selection, but cheaper. And so we wanted to validate this. And so I have this phenomenal student, Holly Lane, who got her master's, and Jose Crosa and some of his colleagues worked with us on this. And we took that data and we looked at how we could predict yield from the grain we harvested from those plots. And we were absolutely blown away to see that under some scenarios, the R squared between what was predicted from this NIR, from the NIR spectra, and what was actually observed for yield. So here in this case, the observed yield is on the x-axis, the predicted yield is on the y-axis. You can see this correlation was very high and would help us predict our top yielders. Now this is different than what we were doing here. So the traditional use of NIR is very much like QTL mapping. Let's develop a calibration for starch, or let's develop a calibration for oil, right? And so we've got thousands of different spectra that we're measuring, both in terms of number of individuals, and we measure something at 8,000 nanometers and something at 8,002 nanometers and something at 8,003 nanometers, and we measure the reflectance values of all of those, except these are wave numbers, not nanometers. And so then we can take all those thousands of spectral measurements, just like you take thousands of genetic markers, and use those to develop a predictive equation. So this looked very promising. When we compared this to protein versus actual yield, it far outperformed protein. It far outperformed our prediction from protein, starch, and oil, because there is a well-known correlation that as you increase yield, you decrease protein. So we wanted to make sure we weren't just picking up on protein for this NIR. Okay. And functional regression, which turns out to be the more powerful approach, actually had better overall results under the right scenarios. Now, what was really exciting is that same year, we had happened to collect a lot of other near-infrared spectroscopy data from other breeding trials. And we found in many cases, the calibration we developed from one set of breeding trials actually worked and predicted another set. But we still do have this open question. Is it predicting yield based on composition? So is it predicting something based on the protein content? Or is it helping us figure out which individuals are related and then helping us find things that look like the highest yielding individual? Okay, so what does the future look like? So this was a benchtop NIR. We still haven't done this completely with UAVs. I showed you Natalia's data, but 530 observations is not like thousands of observations. But where we're going is we want to extract as many different features as possible between the plots. We need to get out of the mindset for breeding that we're going to just measure plant height or vegetation index or something that we can only measure on the ground. We need to figure out how we can measure as many possible things as we can, and then develop the algorithms to use them in prediction. So we're going to do that. We're going to, whatever we can do to extract information from these plots, then we'll test the heritability and use that to predict yield, and that'll help us find new biology. So what does this look like? So this is how my data structure would be set up. This is just a cartoon. We would have our dependent traits of interest like yield. We would have nutrition or ecosystem services or whatever we care about. We can calculate the heritability of those because we have multiple replicates of all these hybrids in multiple environments. We'd also have physiological measures, so like flowering date or leaf angle or leaf number. We'd also have our temporal phenotypic trait measurements, like height on day 30 and height on day 60 and height on day 90. And then maybe we'd also have other things we've never even dreamed of extracting beyond vegetation indices, like texture. I mean, I don't know what texture is, but it's something the remote sensing people talk about. And it's something you and I can see looking at an image, that one has a rough texture and that one has a smooth texture. If we can have an algorithm that extracts that, we can use that in our prediction. And finally, we can have our genomic measures. We can have our SNPs. Right? So we can combine all of these predictive variables into a dependent trait of interest and use some type of statistical analysis like functional regression to predict the best untested hybrid for untested environments. We probably are going to have to calculate a relationship matrix from all these and maybe plug in weather data. But this is what the future of plant breeding to me looks like. And when I look at what costs me the most in my program, it's probably the genomic measures. I can collect UAV measures much more cheaply and easily as these algorithms are developed than I can genomic measures. So if they're more predictive, then at some point I might just get rid of and stop collecting any type of DNA measure. I think I'm going to skip over this. I do just want to point out one last thing in terms of structuring this data. We have this idea of global calibrations and local calibrations. And I've already used the example of biparental populations. So that's a two-parent population, right? So if we have two-parent populations and we develop any type of calibration, this could be a genomic selection calibration, it could be a phenomic selection calibration, it could be an NIR calibration. If we develop that calibration using one biparental population, it's going to have low variance, so it's going to predict all these very accurately, but it's going to have a high bias to this population, so it's going to predict another population very inaccurately. If we use a diverse collection of accessions and the same amount of data, it's going to have low bias in that it'll probably give you estimates that are reasonable for all of these biparental populations. However, it's going to have high variance, so you're not going to have very much specificity. It's going to be the error on all these, or the confidence interval on all of these is going to be very large. You know, it might be 10 centimeters plus or minus 10 centimeters in this case, and 10 centimeters plus or minus 1 centimeter in this case. So it's really important as we think about developing any type of predictive algorithm, again, genomic selection, NIR, phenomic selection, that we choose our material very carefully to reflect what we ultimately want to predict. Now, before anybody gets too excited, I have a little bit of a disclaimer here. This is called the Gartner hype cycle, and most technologies follow this. There's some technology trigger. There's a peak of inflated expectations where everybody says, oh, this is going to be the greatest thing ever. Then there's a trough of disillusionment when we start to figure out things don't work as well as we hoped they would, or maybe they were oversold to us. Meanwhile, there's the slope of enlightenment. That is, people who are still working on the problem are making actual important gains. And then there's a plateau of productivity where these things actually work and are useful. So UAVs in agriculture and high throughput field-based phenotyping is still here at the technology trigger and the peak of inflated expectations. I just told you how great this is. But I expect that we're going to continue to come through these troughs of disillusionment where, well, maybe there's more problems than there are solutions at this point. But eventually, I do believe that that UAVs in agriculture are going to be way up here. I mean, we already know that UAVs are very productive. They were, they really only came on the scene in the last 5 or 10 years, at a commercial scale, and now they're routinely used for lots of purposes. Plant breeding is a very mature, highly productive technology. Plant genomics maybe a little bit less so, right? Precision agriculture maybe a little bit less so. We cover this in our +1 article by Shi et al. On unmanned aerial vehicles. I'm going to skip over that, and I'm going to go to acknowledgements here because I took the full hour I was planning on taking. So a couple key philosophical concepts. Crop improvement mainly improved by predicting the best genotype, not from discovering genes or biology. That is, genomic selection has resulted in more real-world gains than QTL mapping or GWAS. Biological discoveries under controlled conditions tend to rarely make a difference in the field. So if I discover something in the greenhouse and then I take it to the field, it tends to fall apart. That's because there's too much noise in the field. There's too many biotic factors and abiotic factors interacting in it. However, if we discover something in the field, if the signal can outwhelm the noise, it can open a whole new area of research. So dwarfing genes, for instance, there's something so impactful in the field, you can see them, and then you move, they're gonna work. And that can open up a whole new area of research. Field phenomics is a lot like genomics was 30 years ago. And a lot of people are focused on using it for discovery. So measuring plant height, for instance, plant biology or physiology. But it's probably going to be much more important as a plant breeding tool, doing prediction than we ever would have thought. And we can learn that history from genomics. So measuring phenotypes that can't currently be measured is likely way more valuable than just automating or increasing existing measurements. But we have a lot of needs. So I can't just tell a farmer today, go buy a UAV. I can't even tell a graduate student, go buy a UAV. You're going to have great results from it. We need a lot of things. We need new sensors. We need new data handling and storage procedures. We need new analytics, and we need new philosophies like the one I was talking about above. We are still looking at data from 2017 in my program. A lot of the 2018, 2019, 2020 data we haven't even touched because it takes so long to process it. And there really is no single raindrop to blame for this flood, okay? It's just a lot of really small problems or a lot of small unknowns that accumulate to make it to be a little bit challenging to do what you want to do. So this really has been rocket science for most of the groups I've worked with. It's really easy to show you that you can do plant height. It's much more difficult to routinely collect data that can be useful in some type of decision-making in the long run. You really need to have and involve people from other disciplines though. And interdisciplinary going into transdisciplinary or creating whole new things is really hard, but it's what's really needed to solve hard problems. So all in there, there's a ton of people to thank. I tried to thank many of them as we went on. Lots of funding thanks to USDA for the support over the years, to Texas Agri-Life Research and the Texas Corn Producers Board and the Iowa Corn Board for the support over the years. Hopefully this has been enlightening, and I look forward to our discussion and figuring out a way that I can help you and you can help yourself to develop these tools and make them productive for you. So thank you.